hearing from is Desktop Genetics, and Riley Doyle, Chief Executive Officer there, will be giving a short 10-minute talk on their company. We'll also be hearing from Dr. Abel Ureta-Vidal, who is the Chief Executive Officer of Eagle Genomics. Just a few things on the webinar. So the webinar will give a brief outline of what's happening in the bioinformatics sector, how this can accelerate drug discovery, and also hearing from key companies at the heart of bioinformatics, their projects, their platforms, and their main objectives. A couple of things before we start. Just a few housekeeping things. All questions will be at the end of the talk. Please either write your questions on the chat on the right-hand side, or hold them up and I'll be unmuting everyone, ensuring that everyone can get their questions. Additionally, we will be recording the webinar, so if you want to share the webinar with colleagues who haven't been able to join us today, that will be available. If you are on the webinar and you're not speaking, if you can mute yourself to ensure that there's not any background noise, that would be great. But without any further ado, I'm going to pass over to Riley Doyle, our first speaker from Desktop Genetics. Over to you, Riley.
Thank you. Marvelous. So I'm going to talk to you all a little bit about the work that we do over here at Desktop Genetics, and I'm going to try and keep it fairly high level and focus really on the core sort of application of drug discovery, because some of it can be a bit more too heavy on the molecular biology side. So I'll try and keep it as sort of broad as possible. But if you have some more technical questions, my colleague Eddie is going to be around and he can help answer those at the end. So just briefly about me and who this voice in your computer is. My name's Riley. I'm the CEO and technical lead at DesGen, and in addition to sort of my CEO responsibilities, my main sort of focus on the technical side is really therapeutic CRISPR, working with companies like Editas Medicine, the overall system and information architecture for our bioinformatics platform, as well as the application of machine learning to genome editing and CRISPR. Previously, I worked in a bunch of different sort of bioinformatics or engineering roles, such as at Genentech, where I was on the sort of IT strategy working group, and also looking at things in regulatory affairs. So briefly, I just want to give you a quick overview of Desktop Genetics. I want to talk very quickly about CRISPR, specifically how it can help in target discovery and target validation, talk about some of the informatics opportunities here, and then really show you some of the impact that informatics is able to deliver in this space. So Desktop Genetics is based here in London. We are also setting up an office in Boston this year. We do bioinformatics software and services, and in particular, we specialize in the CRISPR-Cas9 system for genome editing that's been all over the scientific press of late. You may have heard of us, or you may be familiar with some of the things that we do. So we have desgen.com, which is our cloud software, largely targeted at the academic market. But then the other thing we do is we work with companies to do more bespoke services where they want to manipulate a very particular genome or conduct some of these functional screens. And so we work with a range of different companies, some on the more synthetic biology side, some more towards the therapeutic side. And I think really this is one of these sort of classic graphs that you always get in every bioinformatics talk, which is that biology is ultimately a data science, and these various omics techniques, in particular next-gen sequencing, has allowed us to really see into cells in detail that was never before possible, and we're generating tons of this sequencing data. And so the need really now is to figure out, well, what does this sequencing data actually do? And to do that, we've had to rely on a couple other techniques, one of which that's come along quite recently is CRISPR-Cas9, which is originally a naturally occurring bacterial immune system that has now been repurposed for doing functional genomics. Effectively, what happens in CRISPR is you have an RNA programmable nuclease that can go in and cause a double-strand break at a particular location in the genome. And then the cell's DNA repair machinery is engaged and will come in and either repair this by correctly, possibly incorrectly leading to a loss of function, or it will patch in a particular new donor sequence, giving you a targeted mutation. CRISPR has then been adapted to also, instead of cutting the DNA, be a way of targeting particular effector molecules to a very precise locus in the genome. So you can bring in activator proteins, repressor proteins, reporters like GFP, and there's even work ongoing now in bringing in epigenetic modulators as well. And what's really cool about these techniques is that you can not only have this sort of specificity, but you also can actually stack them together. So people have been able to do this in a very parallel way where they will sometimes upregulate a gene while knocking out a gene at the same time, and doing this in the context of a high throughput screen. And so that's quite powerful. I can talk more maybe if there are questions about sort of the details about CRISPR, but I think the key takeaway for purposes of target discovery is it's a way of looking at very specific regions of the genome. It's also incredibly popular. There are about 43,000 labs using it today. The amount of hype and publicity around CRISPR has just exploded recently, and we think that it's just going to continue. One of the reasons for this is that it's quite a robust technique, and it works well. Unlike earlier technologies or some similar things like such as functional RNAi screens, is you get cleaner knockouts that are a bit easier to sort of deconvolute and to analyze. And I'll talk about the effect this has on how you actually go about target discovery and validation a bit later. There's a bunch of different applications, as I mentioned, and so this is mostly just if you want to go back and look over some of the other things that you can do throughout R&D. It is a very versatile technique, and we focus on the informatics that underpins the majority of these different techniques. Though I would say that the most popular is to use CRISPR to do targeted gene disruption. So you're trying to do complete gene knockouts and then look at some sort of resulting phenotype in the cell. Now, as I said, for purposes of target identification, what you can do with CRISPR is identify critical regions of the gene or the genome involved in a particular phenotype. Effectively, how you can think about this is that if you have a protein that has a key active site or other functional domain, that functional domain will show up somewhere in the genome. It needs to be encoded somewhere. And that with this RNA-guided nuclease, you can interrogate precise locations in each of these targets throughout the genome. So you can effectively sort of walk through your gene base by base, looking at what's available. And then what you can do is you can read these out in a massively parallel way using next-gen sequencing, because the guide RNAs act like little barcodes. And to give you an example of what this looks like, if you look at the figure below, you will see that there are several different functional domains that were present in this gene, and they saturated this gene with a bunch of CRISPR cuts. And when they got into the ATPase domain, the signal that they're reading out here is that this domain is particularly critical to cell survival. And so this is an example of the type of way you can use CRISPR to identify that a particular functional domain or gene product is essential to, say, cell growth or viability. And you can combine this with other selective pressures such as drugs, repression of other genes, activation of a gene, or even epigenetic modulation to see then, and sort of elucidate the disease biology in a great level of detail and really determine some really cool drug targets. This is actually being done now in practice, and there's been a number of papers coming out recently about screens that have identified new domains, new targets, and sort of new essential genes that could be used potentially for therapeutic opportunities in the future. So there's a couple key ideas that you have to keep in mind when you're doing CRISPR. You have to balance the on-target activity. How well does this guide RNA cut where I want it to? Number two is how specific is it? Number three is where am I cutting? Is it an essential gene? What domain is it in? What's the sort of context? And number three is the sequence quality. Is this a RNA that's going to sort of crumple up into a hairpin, or is it something that will bind and be quite effective? And this gets quite complicated. And so what we've seen is that when you look at a gene, there are lots of guides you could pick. There's quite a large number of them. And what you have to do is you have to come up with some criteria in order to select them, and this gets very complicated. So this is where the software comes into play and why informatics are so important. The informatics help make this a much more tractable problem. So what you can see here is that we've gone and calculated various properties of these guide RNAs and come up with some different scoring rules to allow you to pick the ones that are most suitable for your application. And so the software tools really help you filter out the good guides from the bad and ensure you get the best outcome for your experiment. So what we're building up to then really is what is fundamentally a literal version of desktop genetics. And it's the idea that you can go into your computer and use software to design these experiments and then send them out to a network of partners that you will need to then execute them. The reason the partners are so critical is that something like CRISPR requires know-how, expertise, specialty equipment, and IP rights from a wide range of stakeholders. So for example, we are an Illumina-backed company. They're an investor in desktop genetics, and so we can work with the Illumina sequencing platform to bring in a lot of your genomic data to design the library. We work with companies like Twist to do the oligosynthesis, Transcripted to do the cloning, Horizon for doing the screening, and then we'll do the informatics and analysis. And so what you need in order to execute a full stack is you have to sort of join up all of these different pieces. And that's really the other place where the informatics is quite helpful. It can really help bring all the data across to all these different players. So one of the things that we found with our user base, we've got about 17,000 users who've designed thousands of guides on our platform, is there are some problems that happen. Specifically, humans tend to not be fantastic at designing guide RNAs, and the machine learning algorithms tended to be a bit more effective. And this is something that was really formed our sort of recent research. And so what we've been doing is building out a platform where we will integrate published omics data as well as novel things coming off the next-gen sequencing instruments, annotate this, compile this together, design your guide RNA screen, also do the analysis, and ultimately do the learning to extract out what are the actual druggable targets here. And so the informatics is really what makes this a very efficient and scalable process. And the sort of net result here we've seen is that whereas with manual design guides on some essential genes, you have roughly on average a 25% success rate, both we and other people have shown that machine learning approaches here are able to now get 42, 65% success rate. So you're able to get a much better outcome and a much more effective screen at the end of the day. And that in turn translates into more drug targets. And so if this is something of interest to you, there's two ways we can work with you. The first is we can collaborate and we can put together a screen depending on the different steps in the process that you need help with, all the way from sequencing through to the ultimate screening and analysis. The other way, of course, is you can go to deskgen.com and sign up for an account and play with the public version of the software. It doesn't have all the same features as the advanced internal platform, but it certainly can get you started doing CRISPR today. And with that, we'd be happy to take any questions at the end of the presentation. Thank you.
Great. Thanks very much, Riley. You can hear any more if you've got any questions at the end. I will be passing over now to Abel Ureta-Vidal, who will be doing a talk on behalf of Eagle Genomics. Over to you, Abel.
Thank you, Hannah. Very interesting talk, Riley. Very interesting. So, yeah, I'll pass the slides. You gave me the hand, Hannah?
Yeah.
Okay. So a bit about me. I'm the CEO of Eagle Genomics, and my background started in bioinformatics in the late 90s, early 2000s. I was involved in the Human Genome Project in France, focusing on the chromosome 14 of the human genome at the time, in charge of, yes, I'm sorry? And I was in charge of the pipeline analysis, or bioinformatic pipeline to annotate the chromosome 14 of the human genome and get after the genes. And I joined then, not long after that, in 2001, the EBI in the Ensemble project, and I was in charge of all the comparative genomics aspect of that project. And I left then the EBI in 2008 to found Eagle Genomics. We are based here in Cambridge, UK, at the Barham Research Campus, and we are also a bioinformatics company, like Desktop Genetics. We're focusing on software and services around what we call orchestration of omics data, and not just saying genomics or sequencing data. And we very much were a cloud-first company. We started using cloud technologies in 2009 straight away when it started to emerge in the mainstream. Sectors are around pharmaceutical, biotech, healthcare, and personal care companies, and we have had blue-chip customers from the US and in Europe from the onset of the company. So let me kind of get back to the origins or roles of bioinformatics because this is kind of the subject of the webinar. And very much, the first time bioinformatics won its first gold medal and its recognition to the world was in the Human Genome Project. Before the human reference was available in 2001, the estimation of gene potentials in the human genome was about 150,000. And there were two kind of competing projects. One, the public consortium that published in 2001 in Nature. This public consortium I was part of was doing the old-style chromosome per chromosome clone-based sequencing, while the Stellera commercial initiative that published in the same year in Science was implementing new technologies around shotgun sequencing. And in both, basically, bioinformatics was very, very important. But more importantly, once you had the draft genome, is a re-estimation of the number of genes. It went very, very dramatically down from 150,000 to 22,000 genes. So why is that important? One is showing how bioinformatics was important in analyzing the data. And secondly, it was very important in drug discovery because you can see that the range of potential targets or drugs were down dramatically, and that has a big, big impact in the kind of targets that can be used for drug discovery. That was in 2001. There were big promises for this project to accelerate discovery and develop new medicines. But as you know, it's been 16 years, and discovery really has been stuck and has not moved as fast as people expected. One main reason, I just mentioned that, was the lack of new validated targets. Not all genes in the human genome are, druggable or potential targets. And then because of that, very few new drugs came on the market. There's also pressures from, costing. So there is big pressure to reduce the drug development cost. And also at the same time, pharma is reducing R&D spend in that area. There is more and more competitions. As you know, in the pharma industry, a lot of patents for current drugs are getting to expiration, and there is generic drugs coming on the market. So less profits, less money reinvested in R&D. And the last one is the societal and regulatory pressure for precision medicine. So what does it mean? It means getting the right drug with a proven efficacy in the right condition of the right disease and to the right patient. And that's very kind of getting more and more pressure for pharmaceuticals to get the products out. But at the same time, there is a lot of forces that are pushing for change and to transform discovery. The most obvious one, and Riley made mentioned that a bit, is there is more effective and cheaper data generation platforms. So the sequencing platform in particular, Illumina, is one of them. But we have to forget that there are other platforms for generating other kind of data like transcriptomics, expression, metabolomics, proteomics that are coming as well. At the same time, cheaper access to data storage and compute. I mentioned to you cloud. This has been one of the transformations in our sector. To be able to store data and analyze it, meaning that virtually you can imagine a world where there is storage and compute is free almost. So there is no excuse really to not analyze the data. At the same time, this field requires multidisciplinary collaboration within industry and also externally with academia, and there is eagerness to have that happening. And this has been also accelerated by government priorities. And here's I show you the timeline since 2001 when we had one genome sequence, the human reference, and then little by little, the 1,000 genomes, then the UK 10K here, the Genomics England 100,000 genome sequencing, and earlier last year, kind of the announcement that the US will be potentially sequencing 1 million patients. So a deluge of data is coming our way, and although it feels that it's cheaper to get the data, not everything is cheap. And this was a kind of a little paper published in 2010, 6 years ago, that's still very much relevant, where platforms for data generation sequencing are aiming at $1,000 per genome, and is getting even lower than that. But then there is all the cost of the analysis, and the kind of counterpart was this $100,000 analysis cost. And that's what bioinformatics is supposed to be helping with and coping with the data deluge. But I'm going to give you my definition of bioinformatics. A lot of people believe that bioinformatics is mostly focused on the data analysis, the raw data analysis. But we believe that about 30% is only represented by that. And I think Riley mentioned that bioinformatics is also there to bring data together between different partners to be able to access it. And that is very, very important to be able to scale up data analysis consistently. So we believe that another 70% is what we call the preparation of the data, strategic planning, data curation, and organization of data. And these are basically steps that are required upstream of any effective data analysis. And data analysis, now, what is the outcome in terms of the drug discovery is one is target identification, and more importantly, target validation, but also understanding the mode of action of the drug on the target and the pathways, and designing then kind of the right preclinical experiments that will be kind of used to de-risk then the potential entry of a drug into clinical phases. All these kind of rely on working to assess the data quality and the data relevance to answer a specific question that the scientists have at hand. So very much the end game is here to enable the scientists and the biologists or the bioinformatician to enable what we call the talk with the data. So either the data on their own experiment, but remember, we're in a world that there is not just the data of the scientists, there is the data out there with the collaborators and the data that is accessible publicly as well. And so we are basically all aiming at enabling data-driven drug discovery, which one successful outcome of such an approach would be the ability for a system accessing the right data to generate new hypotheses that the scientists can then test in the lab and generate, again, new data. And that's where bioinformatics is quite important. Here's a slide I want to give you kind of a view of what we believe the trend is in the industry, and I'll get through that through kind of our path since 2008 when we started. Very much, at the time, looking at genomic data, sequence data generated at our customers, and put that in context of the human genome reference. Very quickly, we had to kind of do data analysis and use cloud to scale up large data sets analysis. And very quickly, we also saw that we had to look at coping with the quality of the data, the relevance, and start do data curation. So preparing the data relevant and as a good quality enough to do the analysis. So where we ended up last year was, having the ability to build data catalogs that are relevant for the research of our customers. But one thing that we had to work through is, okay, you have a data catalog, and it's basically the so what. How do I use this data to do maybe CRISPR experiments, as we kind of just discussed here before, or how I use this data to look at expression patterns, or how I look at this data to look at some GWise association studies. And very much what the industry is going through now is looking at what is the way to select the right data, the relevant data for getting a specific scientific outcome or answering a specific question. And that's kind of going up forward, enabling this data conversation that will enable data-driven drug discovery. So that's the path and the game we are in. And if I just finish with this slide that gives you a very, very kind of quick, simple view about where Eagle fits in this scheme and trend, is you have on the left the scientist, the biologist, the kind of bioinformatician, and on the right, all the myriad of data sets that are around that needs to be curated, measured, and assessed so they can enable the conversation between the scientist, the biologist, and the data to make sure the analysis that is then kind of done can give the right answers to the biologist to continue his research. I won't enter into much more details on that, so we have a webinar, Abel webinar coming up on the 25th of February, and there will be more details on our website, but I'll finish with that. Thank you very much.
Thank you very much, Abel, and thank you as well to Riley. We will be now moving on to questions, so I'm just about to unmute everyone. So if you've got any questions at all, please let us know now. I've got a couple here, but I want to see if the audience have any first. Okay, I'll read my questions out first and see if we can get any more along the way. Firstly, I'll start with asking you a question, Eddie. How do you go about incorporating new scoring functions, algorithms from the literature into the platform?
Hi guys, my name is Edward. Can everyone hear me okay?
Yeah.
Great, so I'm the Chief Business Officer at Desktop Genetics. Riley had to duck out to some meetings, so I'm going to answer some questions for you. So the question is, how do we go and incorporate the new scoring functions, new algorithms that are coming out of the CRISPR community into the DeskGen platform? So there's a couple of ways we do this. First, we start with the R&D team and the Genomics Services team evaluating the new paper. So a new paper comes out with a new scoring function, say, for the ability of a guide to not only bind at a target site, but also elicit a cut at that target site. So we'll have a journal club about this. We'll evaluate the paper and the findings. We'll discuss those. And in the meantime, the marketing team will go out and test the demand amongst our user base and our customers for having this new feature incorporated. The reason we do that is in the past, we've seen that the CRISPR community can get very excited about, say, a new ortholog for Cas9, which appears to be very useful, but then subsequently we see there isn't that great a demand for its use in the software. But a new scoring function tends to be far more in demand. So once we agree that there's both a sort of need amongst our user base and there's good data to back up incorporating it into the tool, we begin the long process of porting the new rules, the decision trees, any relevant data into the software. Then we go and we test this on our showroom server for the public version of the DeskGen platform. So we'll test this with a few users and make sure that it's up to scratch. And if so, then we'll release that on the production server, which you can access at deskgen.com. And in the meantime, we also do a confirmation of the new scoring rules on the advanced version of the platform. And what we do is we look at libraries that we've designed historically, so libraries for certain clients, and we'll rescore the performant guides using the new scoring function, and we'll see if there's a correlation there. So were there guides that performed well using old scoring functions, which we didn't anticipate, which subsequently perform well when we rescore using the new scoring function? And so from there, we can do a little bit of analysis and understand if this new scoring function is a significant improvement on the old one, and then we can use that as data just to sort of make sure people are confident in the libraries that they're buying using the new scoring function. So it's a pretty long process, and it can take anywhere from a few weeks to a few months, depending on the nature of the function or the new feature that we're putting into the software.
Thanks, Eddie. Another question here, this one's for Abel. How will the advent of public data accelerate this trend?
Well, that kind of is very much an acceleration factor. There is more and more public data coming in. So in the slide I mentioned kind of the 1,000 genome, the 100,000 genomes coming in. And if you look at data like patient data from the ICGC, which is the International Cancer Genome Consortium, they are sequencing patients on different cancer types, and this data is difficult to access apart from the summary because there are individual information on patients. It's time-consuming and lengthy to access, but what we're doing is making sure that this data is easily accessible. But more importantly, when it's accessible, is making sure that this data has been assessed in terms of quality and relevance for the specific question the scientist is looking to answer. And this is for sequencing, but there is more data coming in publicly around imaging as well, and other kind of omics data sets. So we're focusing very much here on sequencing, but there is a myriad of other kind of data that's coming in. And the scientists at the moment is finding very hard to decide which one they need to look into. And they're looking for systems to give them some guidance on that. And that's what we are helping and working on at the moment at EO.
Thanks, Abel. Next question for Eddie. How is DeskGen's public platform different from the internal one that you use for library design?
Okay, so Riley alluded to this a little bit, but I guess I'll dive deep on this. So the version of the DeskGen platform which you can access on our website is really aimed for the academic field. It's generally used for case-by-case experimentation for the design of maybe one guide RNA to a few dozen guide RNAs. So it provides you with the essentials that you need to do CRISPR gene editing, and specifically guide RNA and CRISPR vector design. And it combines a number of different algorithms from different public tools that have been published by various academic groups. So back in 2013, the only CRISPR design or CRISPR guide RNA design tool out there was the MIT tool from the Zhang lab. And we started with that, we optimized that, we made a few modifications to the algorithm. Then some more scoring functions came out, and we incorporated those into the tool. So you get the sort of whole range of scoring functions in addition to having an integrated genome browser. There's some dedicated modes for knockout and for knock-in, because the things that you need to do to appropriately design a guide or a donor differ depending on your experimental intent. But it's not something which you can really use for, say, high throughput guide design. If you wanted to design 22,000 guides, this is something that we would do for you using the advanced version that we use internally. So typically how this works is you'll come to us with some requirements for a library. Perhaps you want to do a whole exome knockout. And in that case, we'll sit down with you, and we'll have the bioinformatics team discuss the requirements. We'll make use of the machine learning data that we've been building up for the last few years. And then we'll also design the library based on the specific genotype of the cell line or model that you're working with. So we only support the reference genomes on the public tool. But as Riley mentioned, we have partnerships with a number of companies, including sequencing providers. And so we can sequence the cell line that you're working with and design a custom library that makes use of all possible algorithms, our machine learning data, and really creates a library that is specific to the genotype of the cell that you're working with. So this service is primarily used by pharma, biotech, and some of the academic labs that are looking to do larger projects, as opposed to the, say, PhDs and postdocs, which just want to knock out one or two genes, and in which case they'd go for the public platform.
Thanks, Eddie. And we've got a question from Ben Sykes here to both of you. To what extent do you guys collaborate with academics on platform development, or do you view academics as customers and users only? So, Eddie, do you want to go first?
Sure. Yes to both of those things. So academics certainly have been and continue to be customers for both the public platform and our library design services. However, we also collaborate with a number of academic groups. We've got some great relationships with, I guess, the key CRISPR labs in the US. We have some grants which we'll be announcing towards the end of this month as well with a lab here in the UK. So we're always looking to collaborate with researchers. If there's a specific library that you need us to design, then we'd love to work with you on it. A lot of the time we learn a lot about designing some of these weird libraries for, we call it a weird genome, basically a genome that isn't a traditional model organism. And we like to work with people and just see what we can do to extend our technology platform from there.
Thanks, Eddie. Abel?
Yes, in our case, we've been kind of collaborating with academia very much the onset of EGO. We have a strong collaboration with the EBI and the Ensemble platform project. And as far as kind of the kind of data organization platform we are building, yes, indeed, we see academia as, they are the main data producers and data sharers, and they need to be able to provide this data in the easiest way possible so it can be reused by others in their research. So collaboration with academia, yes, we do that, and it's very good for us in terms of understanding the need of the users to make sure we can develop something that is really relevant for them.
Yeah, if I could just add to that, what we tend to see is that the pharma companies, the biotech companies are typically repeating experiments that have been done by academics, whereas the academic researchers are typically doing something very new. So it's an entirely novel use case that we see, which we like to work with people on, so then we can at least make sure that this is ready when it sort of goes more mainstream as a technique. I guess you sort of had that same experience, Abel.
Yeah, exactly. So it's all about kind of trying to avoid the repeating duplication of the same experiment once and once again. And what happens in academia, often the experiment is there, it's published, but then data is very difficult to find. And it will help a lot, pharma companies to be able to access the kind of data and experiment that has been run already in academia. So you basically give them a heads up and focusing on things that are new specifically.
Absolutely. And that's particularly relevant in CRISPR, where there's such a sort of potential degeneracy in library design, and you want to know if that guide has been used in that cell line before and if it worked or not. And knowing that sort of information can really help us from a design perspective, but help our customers, be they academic or commercial, from putting resources into an experiment that wouldn't get the results that they want. So for that reason, one of the things you saw in Riley's slide was this GuideDB, which is an open repository of guide RNA performance data, and we'd love to work with more people on that.
Yeah, because I guess, positive results is good, but, even negative results are good as well, to avoid having to repeat them. That's quite important.
Yeah, and when you're doing bioinformatics, you want to know what worked and what didn't, so you can use that data for statistical learning.
Exactly.
Great, thanks guys. So one more question, and this one is for Abel. Do both biologists and scientists have a continued role here? How will these roles change in a data-driven drug discovery world?
It's not changing. I think what we want to be able to do is the scientist, the bioinformatician becomes a very much super user. And most particularly for the scientists and biologists, how can we make sure the data and the software that is accessible to them makes them forget is an IT and software problem, and is very much something that helps them understand the biology of the disease they are working on. So that's very much what we are working on, and a system that enables them and transforms productivity and effectiveness of their research process. At the same time, as I mentioned, data organization involves data curation, and biologists, scientists, bioinformaticians, are experts in the data they are working on specific experiments, and having a system that can capture this tacit knowledge or expertise is going to be very, very important to make sure a system that we develop is successful.
Great, thanks, Abel. I think that's all we've got time for in terms of questions. But I will be sending a post-webinar email with the recording and also contact detail if you want to get in touch with the companies that you've heard from today. Our next webinar is on the 10th of March, and it's on identifying revolutionary platform technologies for advancing life science research. So do join us for that. Just before we finish, I just thought I'd highlight some of our upcoming BIA events. We have our committee summit in February, which will bring all our committees together in one meeting, Women in Biotech event in March, and Science Finance Dinner and Breakfast in March and April, and then our regulatory conference in May. Just before we finish, I'd like to say a big thank you, firstly to Horizon Discovery, who have partnered with us for this webinar, and to both Desktop Genetics and Abel Genomics for your talks today. They're really interesting, and I hope you have enjoyed this. So, and also thank you to everyone that's listening today. As I said, we'll be sending a post-webinar email out. So on that note, thank you for listening, and goodbye.